we will delve into the TDM of therapeutic antibodies, along with advances in TDM to improve antibody therapies. So why TDM? Because one patient is not like the other, and one dose commonly does not fit all. So when we administer the same dose to different patients, we will likely observe variability in the concentration time profiles between the patients. And this variability can put some patients at risk of toxicity, while others may face therapy failure. And additionally, in the case of anti-infective drugs, there is a risk of antimicrobial resistance development. So it can be challenging to predict whether a specific patient is at risk of overexposure or underexposure. And to address this uncertainty, we can take a blood sample and measure the drug concentration. And if the concentration is too high or too low, we can adjust the dosage to prevent adverse drug effects and to ensure effective concentrations. So individualization of the dosing in this way ideally reduces the variability in exposure between patients. The TDM workflow starts with the administration of an initial dose, often according to the label, according to guidelines or certain patient characteristics like renal function or experience. And after reaching steady state, a blood sample is taken, often at the end of the dosing interval. The concentration is measured and compared to a target range within which the drug is expected to be effective and safe. And if the concentration is outside the target range, the dose is adjusted, often based on nomograms or dosing algorithms or calculations or an educated guess, so that the next TDM hopefully reveals a concentration within the target range. So to summarize, traditional TDM involves the measurement of a drug or other parameter in a biological sample and a subsequent individual dosage adjustment to improve effectiveness and reduce toxicity in a patient. TDM has been recommended for special patient populations like children, for example, and particularly for vulnerable patients, like critically ill patients or patients with kidney or liver dysfunction, or patients with renal replacement therapy, ECMO, or after organ transplantation. And it is also used in patients infected with difficult to treat pathogens that have a low susceptibility, so that we ensure sufficiently high exposure. TDM can be very valuable if the between patient variability in exposure or drug effect is high. And at the same time, random variations in a patient, for example, from day to day, should be limited. Traditionally, TDM has been used for drugs with narrow therapeutic window, with the main goal to prevent toxicity, for example, nephrotoxicity in the case of aminoglycoside antibiotics. But TDM has also increasingly been employed for drugs with wide therapeutic index to prevent subtherapeutic concentrations and therapy failure. For example, beta-lactam TDM is increasingly performed in ICU patients. TDM is further common for drugs with nonlinear pharmacokinetics or for drugs that are prone to drug-drug interactions. And for all these drugs, it is important that the relationship between the dose, exposure, and response is known, and that a target that indicates efficacy or toxicity has been established. And another prerequisite, of course, is that a validated bioanalytical assay is available. Recommendations of TDM exist for various therapeutic areas and also for various patient populations. For example, TDM of anti-infectives in critically ill patients has been recommended for aminoglycoside antibiotics, for beta-lactams, linezolid, teicoplanin, vancomycin, and voriconazole. TDM of immunosuppressives has been recommended for tacrolimus and also for everolimus, sirolimus, mycophenolate, and there is also reports for TDM of cyclosporin. Basically, the TDM process can be viewed as three phases. The pre-analytical phase includes aspects before laboratory sample analysis, like specimen collection and transport, but also the selection of a target you would like to achieve in your patient. The analytical phase involves all steps leading to accurate quantification of the target parameter. And the post-analytical phase involves the interpretation of TDM result to ultimately inform dosing decisions. In the following part of this webinar, I would like to highlight different challenges in each of these phases and also limitations of this traditional TDM approach. It is important to remember that already preclinical steps, or like pre-analytical steps, can affect the interpretation of TDM data and dose selection. So first, it's crucial to use the appropriate sample collection tube, which depends on the bioanalytical method used. The stability of the drug also needs to be considered because for some drugs, cooling of the TDM sample is required to avoid drug degradation and falsely low concentrations. Sampling time is also very important. So traditional TDM necessitates sample collection at steady state, which is reached approximately after 5 half-lives. And this is a limitation if target attainment needs to be ensured very fast, like in sepsis patient, or if in the case of drugs that have a very long half-life. Next, precision in timing is really paramount for this traditional TDM approach. And as you may know, in the clinical setting, the time point at which the sampling was planned often deviates from the time point of the actual sampling. And there was a very interesting study on TDM of anti-infectives that revealed a deviation from the predefined trough sample of up to almost 5 hours, which can ultimately lead to inadequate dosing recommendations. In a clinical setting, the sampling time is not always so flexible. So let's have a look at a hypothetical example. So this patient receives twice daily dosing at 6 p.m. and a.m., and blood samples are routinely taken at 5 a.m., close to the actual trough of the concentration time profile. But if dosing is switched to thrice daily dosing and the routine blood sampling time is kept, a non-trough sample would be taken, which has to be interpreted differently, of course. And an alternative would be to adjust the sampling time to better reflect the originally planned trough value. So these examples show that it's crucial to document the actual time points of dosing and sampling to later ensure the correct interpretation of the TDM result. And it's also important to be aware of the dosing regimen when interpreting a TDM result, because if I measure a trough concentration during intermittent therapy that is shown in black, the target concentration will be different from that of a continuous infusion that is shown in red. It is important to be aware of the adequate target for a patient because sampling trough concentrations is not always optimal. There may be other target measures that are more closely associated with toxicity or efficacy, like, for example, the area under the concentration time curve or the time that concentrations exceed the minimum inhibitory concentration, or, for example, the maximum concentration. For example, the most recent consensus guideline for the therapeutic monitoring of vancomycin for serious MRSA infections recommends a shift from trough-based dosing to area under the curve-based dosing to prevent nephrotoxicity. And here 2 samples are recommended to calculate AUC ideally with a Bayesian dosing software. And here, a PK/PD target, namely an AUC over MIC ratio of 400 to 600 is recommended, with the area under the curve as a measure of exposure and the minimum inhibitory concentration as a pharmacodynamic parameter. It is important to bear in mind that there can be different targets for the same drug. For example, higher targets are sometimes recommended for critically ill patients. And also the target for efficacy may be different from the target for toxicity. For example, for aminoglycoside antibiotics, Cmax has been associated with efficacy and the trough concentration with toxicity. The targets may also be different for different sampling sites and also for different dosing regimens, as we saw before. So after successful sampling, documentation, and transportation to the lab, there are now certain analytical challenges. The accuracy of a reported TDM results relies on a validated assay. And it's important to remember that there may be discrepancies between analytical methods and that concentrations that were obtained using different methods may not be used interchangeably. There are also alternative sample collection methods beyond plasma sampling that may require different analytical methods. For example, dried blood spot samples have small sample volumes, and those might require a different analysis method than plasma. And furthermore, regulatory monitoring and evaluation of assay performance are important to ensure accurate quantification of the concentration. So after determining the concentration follows the interpretation. So in traditional TDM, trough concentrations are typically compared to a therapeutic exposure range. So this approach is rather passive and not proactive. And there are limitations. For example, doses may still be considered adequate even if the TDM sample is close to the boundary of the therapeutic range. With traditional TDM, dosage adjustment usually assumes a proportional relationship between the dose and the sample concentration and linear and time invariant pharmacokinetics at steady state. And sometimes dosing nomograms are used to modify maintenance doses or dosing intervals. And such simple adjustments that really purely use the measured drug concentration are not suitable for drugs with complex or non-linear pharmacokinetics, like, for example, for rifampicin, drugs that exhibit concentration or time-dependent kinetics. And as with traditional TDM, no specific information is provided on how to adapt dosing for an individual patient. Often several cycles of trial and error are necessary to achieve therapeutic concentrations. So we have seen that traditional TDM is a fairly simple approach, but comes with drawbacks. And these limitations have driven the introduction of more flexible strategies like model-informed precision dosing. Model-informed precision dosing uses a pharmacometric model together with a patient's characteristics and a patient's dosing history to predict an individual dosing regimen that can achieve the desired target. There will be 2 upcoming ENOTA webinars that will present these techniques in more detail, one highlighting more general concepts and the second one focusing on software tools. So we hope to also see you at these webinars. TDM has become a pretty widespread strategy to individualize dosing in the clinical setting. But what about scientific evidence supporting this approach? TDM is encouraged in different guidelines. For example, the guideline for managing sepsis that recommends PK/PD-based dosing and also dosing supported by therapeutic drug monitoring. Most published scientific evidence on TDM is based on retrospective studies, for example, implementation studies that compare outcome parameters before and after the introduction of TDM. And prospective studies and randomized control studies in particular are far more scarce. To get an idea of the body of prospective evidence, we have recently summarized prospective studies on TDM and model-informed precision dosing for anti-infectives, immunosuppressants, inflammatory bowel disease, and in the field of oncology. And we found 29 prospective studies up to the year 2022 for anti-infectives, which was more than double as much as for the other areas. So these numbers are not too high. And of course, more well-designed studies are needed to show the benefit of TDM, not only with respect to clinical, but also with respect to pharmacoeconomic outcomes. There are several meta-analyses that have investigated the impact of TDM on outcomes, as shown here for antibiotics. So significant impact has been demonstrated mainly for toxicity outcomes, as you can see here for aminoglycoside TDM and nephrotoxicity. And here nephrotoxicity was significantly less if the drug concentration remained below a toxicity threshold. Another common outcome parameter in TDM studies is target attainment. Clinical and microbiological cure and particularly mortality are far more difficult to show, not least as the sample sizes of TDM studies are frequently rather small. And as you see here, for another meta-analysis, the direction of the diamonds hints towards a benefit of dosing individualization, both for the significant and for the non-significant outcome parameter. The traditional TDM has a pretty long history, but there's also new things on the horizon. And certainly several developments, including pharmacometric models and artificial intelligence, are ongoing. And Inota will not only present 2 upcoming webinars on model-informed precision dosing, but also one on artificial intelligence in TDM and model-informed precision dosing in early December this year. There are efforts to extend TDM to sampling matrices beyond plasma, including saliva or interstitial space fluid, for example, as a common site of infections. And microdialysis can be used for this purpose, which is a semi-continuous technique that allows to measure unbound drug concentrations in different tissues. Dried blood spot sampling is an even less minimally invasive technique that allows microsampling in neonates or in remote or home settings. Biosensors offer the possibility of real-time monitoring, meaning that drug concentrations can be continuously quantified at really high resolution. And such sensors could be integrated into closed loop systems that can automatically adjust the dosing, similar to modern glucose monitoring or insulin pump devices. We have seen that traditionally TDM has centered on measuring concentrations for drugs with high pharmacokinetic variability between patients. But of course, variability also extends to drug effects, which makes pharmacodynamic targets an attractive option for TDM as well. Some biomarkers are already used for dosage adjustments, for example, neutrophil counts to monitor chemotherapy, or in research, kidney-specific proteins to assess nephrotoxicity. There are also further markers that are being investigated as pharmacodynamic targets, including markers of inflammation, such as C-reactive protein or procalcitonin, or markers indicating immune response or severity of illness. And last, TDM is not restricted to small molecules, but it's also conceivable for other therapeutic modalities, such as antibodies. And with that, I thank you for listening, and I hand over to my colleague Erwin, who will talk about advances in the TDM of therapeutic antibodies.
Okay, thank you very much, Iris. That was a very insightful presentation. Let me try to share my screen. I think that's working. I think you should be able to see my screen. Oops. Yeah, sorry. Okay, so let me please start. So there was a very nice presentation, Iris, and it was really about anti-infectives. And I think it's very interesting because now I will be talking about TDM of biological drugs. And I think you will all see some similarities, but maybe also some differences. We're happy to discuss these at the end of this webinar in a discussion. So if you have questions, feel free to drop them in the chat to keep them and bring them up in the end. So when talking about TDM of biological drugs, I'd like to go back into time a little bit with you. 25 years, actually. That's 1999. And that is the time that the European Medicines Agency authorized the marketing of infliximab, a monoclonal antibody, an anti-TNF antibody, for the treatment of a number of immune-mediated diseases: Crohn's disease, rheumatoid arthritis, ankylosing spondylitis, psoriatic arthritis. And it was really just a few years later, in 2002, that the company, Centocor, what is now Janssen Biotech, published this paper. And as you can see, it discusses the relationship between serum infliximab concentrations and clinical improvement in patients with rheumatoid arthritis. And in this paper, there is this one plot where you can nicely see that when the infliximab concentrations increase, that you have gradually more patients who have better therapeutic outcomes. And of course, the other way around as well, that the patients with the less favorable clinical outcomes are more clustered in the quartile with the lowest infliximab concentrations. So this is a very interesting, one of the first descriptions of the relationship between infliximab exposure and response. And in this paper, the conclusion was that some patients with rheumatoid arthritis might benefit from higher doses or shorter intervals of dosing. So this paper really opened the door for dose optimization of infliximab based on concentrations. Then 1 year later, there was this other paper, this time not in rheumatoid arthritis, but in Crohn's disease. And this was published in the New England Journal of Medicine. And I think that already highlights how important and how clinically relevant this research was considered at the time. And in this paper, this is not a company study, this is from data here in Leuven, actually, some former and some current colleagues of mine. They showed that in patients who had infusion reactions, that these patients generally have very low infliximab concentrations. And they also showed that this was often due to immunogenicity. So when talking about biological drugs, we cannot ignore immunogenicity, the development of anti-drug antibodies against the drug, resulting in a higher clearance, low levels, infusion reactions, and also poor outcomes. So these papers were really the start of TDM, by showing the relationship between infliximab exposure and adverse outcomes, but also therapeutic outcomes. Now, if we make a big jump, about 20 years further in time, close to today, I think this is the most recent, very relevant paper on TDM of biological drugs, the results of the Nordram trial, showing that there is a benefit of doing TDM during the maintenance treatments of infliximab. So still infliximab, 25 years later, we're still investigating TDM of infliximab. And in this paper, they show the benefit in a number of immune-mediated inflammatory diseases. Not just IBD, not just rheumatology, also dermatology, psoriasis. And this little plot summarizes actually everything in this trial, that if you do TDM, clinicians are able to keep patients under control, the disease under control in patients for a longer time. So this was really an important study showing the benefits of TDM. So what happened in these 20 years between, first of all, showing exposure-response relationships and today bringing this into the clinical practice by doing TDM? So for that, we need to understand what TDM is in a practical way. And I think we can define TDM as really taking blood samples to measure concentrations and comparing these concentrations with a certain target concentration, and then simply adjusting the dose, or maybe not adjusting the dose, at least in order to achieve or maintain the target exposure. I think that's TDM, measuring, targeting, and then dosing in order to reach the target. And we talk about TDM, and TDM is actually a very old term from dating back to a time when the real challenge was to measure these concentrations. So TDM, therefore, today maybe sounds a little bit passive in the sense that it's really just about measuring, monitoring, and it's not about dose optimization. So that's why some people also suggested that we should talk about concentration control, which is really about controlling the concentrations, controlling the exposure, which is already a little bit nicer, I think. But in the end, it's all about dosing interventions. So that's why some people also talk about target concentration interventions, which is, I think, a really nice term to stress that TDM is actually an intervention. It's about dose optimization. And sometimes this can be done by using models, and I'll come back to that. And then more recently, another term was introduced, model-informed precision dosing. So we have quite a broad spectrum in terminology, but I think in the end, all these terms shed light in a different way on the same process that is TDM. And when talking about TDM, I like to compare it to driving a car or to riding your bike, because it all comes down to steering the wheel, to use the brakes, to accelerate, and that's the dosing. And of course, how you drive your car, that all depends on the setting on the road. And of course, you adjust your driving style based on the setting, because the end goal is to reach your destination, and you want to do that in a safe way. And TDM is not different. If you dose a drug, you want to dose it while using the information about the pharmacokinetics and the pharmacodynamics. You need to know the setting so that you can bring your patients into remission or keep the response. And you don't need insight into this setting if the setting is very predictable. And that's where the problem is. For these biological drugs, the PK, the PD can differ a lot between subjects. So you can just say that TDM is really just a way to get insight into the setting of the pharmacology of the drug in every individual patient, so that the clinician can dose in a more personalized way, trying to bring the patient into remission. So we have to take into account all this variability, what's going on behind the scenes. Now let's make TDM practical. How have people been trying to do that? In the past, and actually not so long ago, still today, the algorithms for dose optimization are relatively simple. And you can see that here, this is what they did in the Northern trial. And I simplified it a little bit so that it's more clear. So you can see that when the measured infliximab concentration is below the target, the algorithm simply suggests to increase the dose or to shorten the interval. And when the concentration is above the target, then you can dose de-escalate by lowering the dose or extending the interval. And it was not different for another clinical trial, the TAXIT trial, another big clinical trial on TDM of infliximab in IBD, inflammatory bowel diseases. And here it was a very simple analog algorithm, a flow chart, allowing clinicians to make decisions on the dosing. There's one big problem about these algorithms. They are very, very slow. you just know that your concentration is below the target, you know that you need to dose escalate, but you don't really know if this one intervention is going to be enough. So maybe you have to do 2 and 3 or 4 interventions to bring the concentration at the target. And that can be a slow process. And it actually shows very nicely from this plot from the recent Northern trial. You can find that plot in the supplementary files. And you can easily see that for the TDM arm, it really takes time for the concentrations to cluster within the therapeutic window. And at 1 year, there are still many patients who don't have infliximab trough levels within the therapeutic window, which is, by the way, a very broad therapeutic window, 3 to 8. So although this trial was positive, was showing a benefit for TDM, not all patients hit the target. So I don't think we can talk about precision dosing here. And that's a problem. And that might also be one reason why several TDM trials failed to show a benefit of TDM in the past. So on the left, and this slide really summarizes all the evidence that we have today for TDM of infliximab. And I'm only talking about infliximab because for all the other monoclonal antibodies, there is even less evidence. So what I'm showing you here is actually the best that we have for infliximab. 3 trials showing no benefit for TDM. On the left, Taxit, Tailrix, Northern A. And on the right, we have a Danish trial and the more recent Northern B trial. And you can also see that it's really this oldest trial in 2014 that really made TDM a very hot topic in the field. And after that, all these negative trials came that were not able to attain the primary endpoint. Nevertheless, we learned a lot from these trials. But the interest in TDM disappeared a bit. And more recently, the interest, thanks to Northern B, was again there. Okay, so now, of course, clinicians need to do things with all of this information, and that's not easy. Of course, we have different types of information, and as you can see all around the world, clinicians developed consensus statements, and you would expect that they all agree, which is not the case. Many different reasons why. One of these reasons is, for example, when the consensus statement was published, some of them take into account Northern B, some others not, they're older. But I think the overall belief in the clinical community is that there is really a benefit for TDM, and we're only seeing that in recent years. So you might assume that all the problems are solved now, and unfortunately, I can show that here that's not the case. So what are we looking at? Here we see for a number of pivotal trials, the remission rates in the intervention arms on the left for many monoclonal antibodies, always for ulcerative colitis here. And you can see that the remission rates for all these drugs in patients with UC are actually very, very low. Infliximab is doing the best here, and you can see around 1 in 3 patients actually reaches a remission. And for other monoclonal antibodies, the remission rates are even lower. So although these are very powerful drugs, they are certainly not powerful enough to treat every patient. And this is what we call the therapeutic ceiling. These drugs can only do that much. And we are using TDM to break this therapeutic ceiling. And now it shows that we can, but TDM is not going to be the only solution. Clinicians are also trying combination therapies of monoclonal antibodies. They try to predict which patients will respond best to what monoclonal antibodies. Still, many clinicians also really just switch. If an antibody fails, they just go to another one because we have several options now. So TDM is just one option. And I think it's important for us to think very well about how we are going to investigate further in the future TDM in order to make it show its full potential. And we've been discussing this together with some colleagues in the IATDMCT, the International Association of TDM and Clinical Toxicology consortium. And a few weeks ago, we were able to publish our consensus statement on TDM of infliximab. And I would like to guide you through this very briefly now. We actually highlight 5 points of attention. And the first one is we need to focus on the right patients. Who are the patients that will benefit most from TDM? Also, it's important to think about the target. What are we actually aiming for? And I'll come back to that. What data should we use to do TDM? I've been talking about trough levels, drug concentrations. I'll give you a few other considerations in a moment. Number 4, how should we translate all this information into actually actionable recommendations for the clinic? And finally, even if we would know that TDM has a benefit, how are we going to implement this in clinical care? So let's go through these points smoothly now. First of all, the patients. What patients should we focus on? I think it's clear from the clinical trials in the past that not all patients really need TDM. Many patients just do fine with standard dosing. So it is important to identify the vulnerable patients. Who are they? Well, first of all, first scenario is that we should try to avoid primary non-response. That means that when a patient is sick and we start treatment, we can use TDM to bring the patient in remission. This is healing, trying to heal a sick patient. And in this specific setting, we should focus on the patients that are the most sick, patients with a high disease activity, for example, patients with acute severe ulcerative colitis, a very specific setting that people all around the world are focusing on today. These patients might benefit most from TDM. Also, patients that simply have a lower sensitivity to infliximab, for example, patients with fistulizing perianal Crohn's disease, another very specific population, but also patients with certain polymorphisms that are linked to an increased clearance of infliximab. These patients might need more close monitoring. And eventually also patients with a young age, just because they have a lower body weight, and it is known from actually the original drug development that the standard dose of infliximab is actually not enough for these patients. So my point is we should identify the vulnerable patients. And there's different scenarios. For example, also when we are going to dose de-escalate patients, we might want to use TDM to avoid that patients relapse. Same for when we withdraw an immunomodulator that can have an impact on the PK of infliximab, we might consider doing TDM here. And one third scenario that the clinicians propose is doing TDM to avoid immunogenicity. And this is especially interesting when patients are restarting the treatment after a drug holiday. Okay. A second aspect, what are we aiming for? What is the target? I've been talking about trough levels, and here we see a nice overview. This is clearly a simplification. It is way more complicated than this, but this is a common average, a slight consensus of everything that is out there today. And as you can see, there are windows, which is interesting. There's no specific target. There's always windows, also indicating a little bit the uncertainty about the targets. And then for some settings, there isn't even a target. Now, honestly, we don't care about PK. We don't care about exposure. We don't care about the concentrations of the drug as such. We do care about them because we know they are related to outcomes. So we're actually focusing on surrogates. And maybe before I move on to the actual clinical outcomes, we've been talking about trough levels, but more recently in a European Public Assessment report, it was indicated that for the subcutaneous infliximab formulation, the dose was defined based not on a trough level alone, but also on an AUC, an area under the curve, the overall exposure, indicating that maybe we are looking at the wrong thing. Maybe we should also consider an AUC, just like my colleague Iris showed that for some antibiotics, we're doing this as well. Currently, we just don't know. So that's an open question. Yet it is generally considered that the trough level is the driver of efficacy of monoclonal antibodies. And recently, and this is just a side note, we developed an algorithm that can guide clinicians by doing TDM in the switch from IV to sub-Q infliximab. By sampling before and then from some simulations, we can actually allow clinicians to predict what the level is going to be after the switch. So this is an opportunity for TDM as well. And as I mentioned, we don't really care about trough levels, we care about outcomes. And then we have many options here: clinical outcomes, steroid-free remission, endoscopic remission, less hospital visits, productivity, many things. And this is a little bit out of the picture when we talk about TDM. When we talk about TDM, we always talk about trough levels. And I think that's a mistake. I think in the future, we should really focus on the link between trough levels or exposure at least, and these relevant outcomes, so that hopefully then we can do better TDM. And clinicians are doing this these days. What data should we gather? Well, it's clear we should gather data on trough concentrations. We very recently did a survey. It was coordinated by my colleague Celia Skele, an initiative from Inota, to investigate how people in Europe are doing TDM of monoclonal antibodies. And we can see here that the ELISA assay is widely used, usually commercial assays. But there's more to TDM than just doing ELISAs. You can do microsampling, which means that patients can sample at home, which gives more flexibility. We can do point of care testing. We have assays with a very short turnaround time, so you can act very rapidly. And we can also measure biomarkers like fecal calprotectin, and we can also consider these in the TDM algorithm. So we should have a more open mind and think about all the options that we have when we do TDM. There is more than just a trough level. How are we going to provide dosing recommendations? That's the fourth aspect. I already mentioned it, and Iris did it as well. We can use models, POPPK, POPPK-PD models. And this is just an illustration, a software tool, TDmetrics, that we developed together with our colleagues at the University of Hamburg to do TDM, model-informed precision dosing effect of infliximab. Just an illustration. This is way faster than the analog algorithms. And actually, the benefits of model-informed precision dosing of infliximab have already been shown in a clinical trial, the Precision trial in Amsterdam. That's what we see here. So you can see that patients on MIPD, after 1 year of follow-up, have better clinical remission rates as compared to patients that did not have those optimizations. So we do have evidence in favor of MIPD of infliximab. And that's why I really believe that MIPD is the way forward to make TDM, TCI, concentration control more efficient. The options are there today. Several software tools are available, so nothing is stopping us from actually trying to do this. And then the final point I'd quickly like to highlight, the implementation in clinical practice. There is a lot to say about this. There is the pharmacoeconomical aspects, of course, the workload. One thing I'd like to highlight is that if we would use these software tools, that it would be great if they would be integrated into the electronic health records so that there is an automatic data transfer and it will make the life of the clinicians easier. And this is just one publication, very nice publication from colleagues in Cincinnati, describing how they developed and integrated a precision dosing software tool for infliximab into their own hospital, into the electronic health record system. So I think with that, I'd like to end. And my main point is that TDM is really just an opportunity to go from equal healthcare, where we give the same dose to every patient, to more equitable healthcare, where we give different doses to different patients, focusing on bringing all patients into remission, giving the most optimal chances to all patients. Thank you for your attention.
Thank you very much, Erwin, for this very informative overview of TDM of antibodies. And before we proceed to the discussion, I would also like to give you some more information on our second speaker. So Erwin is an assistant professor at KU Leuven in Belgium, where he leads the pharmacometrics research group. And after his PhD in pharmacometrics, he worked at the pharmacometrics research group at Uppsala University and also at the Integrative Pharmacology Laboratory at the University of San Francisco. And his research at KU Leuven is focused on optimizing drug dosing in vulnerable patient populations and model-informed precision dosing of monoclonal antibodies, anti-infectives, and immunosuppressives. Erwin works as a scientific expert for the European Medicine Agency, and importantly, he leads Working Group 2 of the Inota Cost Action.
Thank you very much, Igor. So I think with that, the floor is open for questions, so feel free to drop them in the chat or to turn on the mic, turn on your webcams. And we're happy to take your questions.
Well, hello, Erwin. This is Nick Holford. Can you hear me?
Yes.
Perfectly.
Yeah, so I'd like to thank you very much, both of you, for your talks. Really nice presentation. I have to say that I'm still awake. I say that because I just got off the plane coming from New Zealand yesterday, so I'm still trying to adjust to being in Europe. The one question I'm going to focus on, there are many, but one of them is about the choice of a target being the trough concentration. I know when you rightly raised this as a question, but the answer is already known, but it's known by scientists. It's not known by clinicians. A scientist, a pharmacological scientist, would know for sure that the trough concentration cannot have all the information about how that drug is going to work in the patient. The trough is just measured by tradition. It's just because I'm probably the oldest person in this audience. When I was a young doctor, people measured trough concentrations because they didn't know what to do. So it was just easy to remember, try and take it before the dose. And that has kept going, and nothing has changed in 50 odd years, because the people doing it never think. So, and the proof is that they don't think is they continue to measure trough concentrations and believe that what that is also the target. And that's a big mistake, to believe the target is the same as what you measure. And that can be avoided by science and what you call model-informed precision dosing, which I would call target concentration intervention. Those terms that have models behind them, you can make a measurement of a trough concentration and you can predict the clearance, and therefore you can predict the dose required to achieve a target AUC. So that's science. I don't think either of you said those words, that science can actually work. You basically gave your empirical talks, trial and error. That's fine. It's a good start. But from now on, I hope we can move to a science-based discussion. And the questions obviously are, well, what is the exposure? What is the AUC to start with? Not the trough concentration. So that would be a question for this group or for any group working in this area, which very few people address. It was addressed for vancomycin, and thank God we got it right for vancomycin. It's probably true for all drugs. The AUC will be a better target than the trough. Thank you.
From my perspective, I will briefly say something. I fully agree with you, of course. I think in this presentation, I really wanted to focus on the practical aspect, of course. And I fully agree, clinicians, for monoclonal antibodies at least, have been simply sampling a trough right before the next infusion, because that's simple. And if I ask the clinicians I collaborate with, please sample more, I really have to defend myself. I really have to explain why, because it's a big hassle, of course. So I think we can also not ignore these practical aspects. And we have to accept a little bit that even though as a scientist, we know that theoretically we can do better, we have to deal with the practical limitations, of course. That's a side note. Thinking as a
Erwin, just to come back, that trough concentration can be helpful to predict what the AUC is. You know?
Yep.
It's just the measurement. You're confusing the target with the measurement. You can have the same practical measuring a concentration, a trough, and I don't care if they get it wrong, because even better if they get it wrong, because it might be closer to the middle of the dosing interval, which is a better time. But the practicality is not changed. What changes is using science and not clinical practice.
Yeah, so that brings up two questions, basically. What's the correlation between an AUC and a trough level? Overall, it's pretty good, but it's not perfect. So there is a difference. And then there's also the mechanistic question. Maybe you can say me a bit more about that, specifically for antibiotics, because there is, of course, a lot known about different types of antibiotics. And for some, the AUC is important. For some, the trough level is important.
Yes, I also agree, Nick. So, I mean, if you wanted to calculate AUC with what we call now, or what's this webinar is based on, traditional TDM without any model, that of course becomes even more difficult, and people are even more hesitant to calculate AUC. So the AUC based dosing, that will be more the focus of the upcoming webinars, that really then explain the benefits of model-based TDM target-controlled interventions. Of course, it will facilitate science-based TDM a lot, also for clinicians, because then these apps, of course, are more user-friendly than calculating AUC with these traditional two-sample-based methods.
I think we should definitely also consider the mechanism behind it, of course. It is generally believed that you just need to keep your concentration above a certain level for monoclonal antibodies, and that's enough. And it's generally believed that is enough for keeping patients in remission. But if you have a sick patient, probably that's at least a hypothesis, the overall exposure, the AUC is more important. You need maybe a high peak, or you need just more drug in a shorter time. So we should definitely consider the mechanism as well. And I think, unfortunately, 20 years, 25 years later, we still don't really know for these monoclonal antibodies. So there is indeed research to be done. And I think the opportunities are definitely there, especially because when you use models that CE has highlighted. I hope that was a bit of a satisfying answer. I am quickly going to the chat because I see there's another question. Let me see. So the question is it possible to increase the prediction performance of model-informed precision dosing software tools by using artificial intelligence? We could probably expect this question. It's a very good question. I think there's a lot going on that. I don't know, Iris, do you know things going on with AI?
So I'm aware of several examples in the area of immunosuppressive drugs. So a group in France developed a machine learning tool and also implemented in a Shiny app. And they showed that these machine learning based AUC determination and then those predictions were more accurate than conventional model-based dosing regimens. So I think there were publications on tacrolimus, and I think also of mycophenolate. And in this publication, you also find the link to the app. And in their case, it seems to have performed better.
Okay.
But there's not so many examples I'm aware of out yet. There's also some hybrid methods combining model-informed precision dosing, PK/PD-based dosing, and machine learning, for especially when you have kind of outlying patients with very high concentrations that cannot be captured by models. So how to then how to deal with them?
No such thing. There's no such thing as a concentration that can't be captured by a model.
Or like the individual prediction depending on the patient characteristic and then the magnitude of variability. You would have to then tweak the variability parameters, maybe.
An outlier in the normal distributions of the variabilities. Yeah, I think that's something that the group in San Francisco, Insider X von Kaiser, has been working on. So that's indeed, I think MIPD is very promising. I mean, the term itself says precision dosing, but we also have to be a bit realistic. If we look at the performance, it's usually not as good as the promises. So I think there is still room for improvement. I think indeed AI has a potential. The proof is still in the pudding, I think, in the end.
Well, I would like to know how AI can predict the dose. Model-based methods have pharmacokinetic models that can predict what dose is required to achieve a target concentration. That's science. Artificial intelligence, unless it has a model, like a pharmacokinetic model, how can it link what's known to the dose that's required? Now I don't know how that question is answered, or if it is answered, but it is getting to the dose is what's important. One of the problems I'd say with precision MIPD, it's very hard to find out how the dose is predicted. I've searched the literature completely, but I've asked the question of many people. I've looked at key reviews on MIPD. Nowhere does it say how the dose is calculated. They tell you how they calculate the clearance, but they don't tell you how the dose is calculated. So I don't like the term MIPD because they don't actually predict the dose. It's done somewhere behind the scenes, but no talking about it. And that's one of the things I can talk a lot about how to predict the dose. MIPD doesn't do that, and it's not published how to do that. So those who do MIPD, please say, how do you do the dosing?
Yeah, you're fully right. You're fully right. So I think MIPD is then more of an umbrella term, and there is probably different things going on that we don't know, and there's different ways of doing it. And I think the same applies for AI. It's an umbrella term. There's a lot of ways of doing that. But I'm not the expert. I think in a few months we have a dedicated webinar on AI tools. So very welcome there. Absolutely. Do we have more questions from the people online? Thoughts?
So Erwin, I think there's another question in the chat for you. Let's see. So can you comment on different responses in exposure response models?
Yeah, it's a very good point. If you look into the literature and the existing exposure response models of, I'm talking about pharmacodynamic models, mixed effect models for biological drugs in chronic inflammatory diseases, the responses are usually CRP, which is biological response, I guess. So it's not a real response, it's a biomarker, it's a surrogate for response, so not really good. Fecal calprotectin, which is a very good biomarker, widely used, very predictive of what's going on in the bowel. And then what we did is we built a model where we link infliximab concentrations to fecal calprotectin and then fecal calprotectin to endoscopic remission. Endoscopic remission, at least for IBD, is considered currently the most important clinically relevant response or outcome that clinicians focus on. But it's a good question. I think also here we have not been focusing enough on doing these things, on investigating these things. Thank you, Katharina.
Thank you. Thanks.
Good. I think maybe one final question for me for Iris. Do you know about PK/PD or exposure response models for antibiotics? As far as I know, it's always just PK and PK/PD target attainment.
I'm not aware of any routinely used one yet. There's research, but so there's more success examples, for example, in oncology, where they monitor neutrophils, for instance. There's also model-based therapeutic drug monitoring in to monitor the toxicity. But with anti-infective dosing, I'm not aware of routine applications of that.
No,
not yet.
I can give you one example that is very routine.
Yeah.
Measuring a biomarker in order to adjust the dose, and that's for warfarin.
Oh, yeah.
Where the international normalized ratio is measured and is traditionally adjusted by various algorithms, but it can use concentration guided dosing or TCI. And I'll just say we just published that this month in CPT. I mean, it's been done before, but we showed that you can use target concentration intervention to improve outcome with warfarin, as just measuring the biomarker without measuring what's driving the biomarker, the concentration of warfarin.
They're still much more common outside the field of anti-infectives.
Yeah. So anti-infectives, there are work, as you say, I think there's a lot of work being done, but it doesn't get translated into action. A lot of stuff in test tubes with bacteria and antibiotics. A good friend of mine, Jürgen Bulliter, you may know him or may know of him, has spent his working life doing that, but he still hasn't produced anything that works in the clinic, as far as I know.
Cool. Thanks for sharing that, Nick. Okay, good. I see we are beyond 8 p.m. in Belgium. I think unless there's burning questions, we can end this webinar here. Thank you all for attending. The webinar is recorded. The recording will be made available later on the Anotta website. And apart from that, I'd like to wish you a very nice day, evening, night, and of course, welcome you again in a month for the next webinar. Thank you very much. And thank you, Iris, as well, for the very nice presentation.
Thank you very much. Hopefully see you at the next Anotta webinars.
Bye-bye.
Bye.